Welcome back, students, to the third lecture of regulation of gene expression. In this, we're going to discuss regulatory proteins. We'll get an idea of the basic structure of regulatory proteins, various examples, and we'll discuss the examples in detail individually. What does a regulatory protein do? It goes and binds to the DNA at a specific position, interacts with other regulatory proteins, and causes either turning on of the transcription or turning off of the transcription. In order to mediate that effect, it should be capable of binding to DNA. So it should have a DNA binding domain, and it should be capable of binding to other proteins. For example, RNA polymerases interact with other proteins and other regulatory proteins which interact with the RNA polymerase or others. For example, the CAP protein binds to CRP as well as RNA polymerase. So the regulatory protein has to have DNA binding domains as well. The adjoining figure just gives an overview. This is a representative diagram of a regulatory protein. There is a DNA binding domain represented at the bottom in light blue color. The blue colored arc shape portion represents ligand binding domain, and the regions marked labeled 1, 2, 3, 4 represent activation domains. So the DNA binding domains, it's a common pattern that the regulatory proteins dimerize in order to be capable of binding to DNA for so many regulatory proteins. So the regulatory proteins, in addition to having a DNA binding domain and a protein binding domain, also need to have an additional domain, which is dimer forming domain. Also clear from this structure and your knowledge about the protein structure, a domain is, and the DNA binding domain hence, is only a very small portion of the whole of the 3D structure of larger structure of a protein, just as in case of enzymes. The sequence to which the DNA binding domains bind in DNA are specific, so it ensures that only certain regulatory protein goes and binds to only certain regions in the DNA. These sequences are generally inverted repeats. You also know them as palindromes. The DNA binding residues in the protein are the basic amino acids. Why basic? Because they need to interact with the DNA, which carries negative charge because of the phosphates, while the protein binding domains of the regulatory proteins has to interact with the protein. So the amino acids present there are hydrophobic amino acids. Let's just discuss various types of domains present in the regulatory proteins. These are the few examples. The first one here is helix-turn-helix motif. It is present in E. coli, phages, and mammals. The common examples of regulatory proteins having helix-turn-helix motif are lac repressor, CAP, and the homeobox proteins of the mammals also consist of helix-turn-helix motif. Then is zinc finger domain. Zinc finger domain is present in E. coli, yeast, Drosophila, Xenopus, and mammals. The common examples of regulatory proteins having zinc finger domains are transcription factor 3A, and the proteins of steroid receptor family also consist of zinc finger domains. Then is leucine zipper motif. These are present in yeast and mammals. The common examples of regulatory proteins having leucine zipper are FOS, GIN, CRE binding protein, and SAMIC. We will now be discussing these domains individually, and in addition to these, some other few examples. So helix-turn-helix motif was the first domain to be discovered. As the name suggests, it has one helix turn, and then helix. And you can also see in the adjoining figure, the domain, the binding motif, has around 20 amino acids, of which the 6 to 7 amino acids form alpha helix, first 6 to 7 amino acids. Then these are separated from the second alpha helix by a small turn, a beta turn. We know that a motif is a smaller part, so this portion, the helix-turn-helix motif, is the reactive portion of a larger protein. Of the two helices, only one plays role in the interacting and binding to the DNA. So it has specific interaction, sequence specific interaction with the DNA sequence to which it binds. You can see in this diagram. It is a very common motif in case of prokaryotes. Lac repressor is one example. It is also found in eukaryotes though. So the second example we discuss is helix loop helix motif. Uh, you may feel that it is the similar motif, just one word has been changed, the loop and the turn, but you'll get to know how the two are different. It has two short amphipathic alpha helices which are linked by a loop of variable length instead of the turn. So this is the example of one motif. You can see one alpha helix, then yellow colored, you can see the loop, and then the C-terminal second alpha helix represented here in the green. This diagram itself should make you clear what makes the two motifs different. So it has the N-terminal alpha helix, which has basic residues. So basic residues means it goes and binds to the DNA, then the middle loop, and then the C-terminal region, which interacts with the protein. So it has the it has the hydrophobic amino acids. The structure of the helix loop helix motif indicates that this one alone may not be able to interact with DNA properly. So there has to have two such helix loop helix motifs of the protein which need to come together to form a functional dimer. So now it's clear for you that while helix-turn-helix motif had a single polypeptide forming the whole structure of the motif, the helix loop helix motif has two separate polypeptides forming the functional motif. So when the two separate polypeptides are involved, it can either be in the form of a homodimer or a heterodimer. The next example is leucine zippers. If you want to go ahead and define, a leucine zipper is a DNA binding structural domain having hydrophobic amino acid leucine at every 7th position in sequence, forming a straight line along the surface. You can see here in the joining diagram that this portion, in the bottom portion, is the leucine zipper, which the polypeptides are intertwined, and in the inside picture, it is shown that how the leucines are aligned towards the surface and they are interacting with the other leucines. So the leucine zipper in itself does not form the portion of the protein which interacts with the DNA. It only holds up the DNA binding portion intact, providing a proper pseudochemistry. So again, the leucine is a hydrophobic amino acid. It cannot interact with the DNA. So the portion which binds to DNA has to have basic amino acid, being any one of the arginine or the lysine. It was initially understood, or I should say regarded, that leucines interdigitate like a zipper as shown in the diagram here, but instead it is now understood that they are lined across each other as represented in this diagram in the red color. So the leucines of the two different motifs, the two different polypeptides interact with each other line to line. So it is a very common motif found in eukaryotic transcription factors, whereas it is relatively less common in case of prokaryotic transcription factors. The next example is zinc finger motif. The zinc finger motif has about 30 amino acid residues and they form an elongated loop, as you can see in this diagram. The loop holds together, it is held together at its bottom or at its base by the coordinate bonds with 4 of its amino acids towards the zinc metal. So the zinc ion forms the bonds. The 4 amino acids which are involved in the coordinate bonding are either cysteines or 2 cysteines and 2 histidines. So the configuration remains 4C or 2C2H. So in this case, zinc does not go ahead and interact with DNA. It only stabilizes the structure of the zinc finger motif. So the binding of one zinc finger is not sufficient to keep the protein bound to DNA to which it is to a DNA sequence. In order to have an effective binding, a stronger binding, the regulatory protein which has zinc finger motif has multiple zinc finger motifs in it so that all of them coordinately bind to the DNA and hence there is a stronger, substantially enhanced binding of the regulatory protein to the DNA. So the most common configuration which is observed in case of humans in zinc finger motif proteins is C2H2 instead of C4. The next example and the last example we're going to discuss is homeodomain. So it is found exclusively in eukaryotes. It is structure related to helix-turn-helix motif. This is the representative diagram of the helix-turn-helix motif. And the motif has 60 amino acids. Now, one would say that why do we have a separate name and a separate class for a motif which we've already discussed, but since it is very significant, it has to be discussed separately. It is found only in eukaryotes, not in prokaryotes, and it was first discovered in homeotic genes, hence the name homeodomain. You must be aware the homeotic genes are the ones that regulate the development of body patterns. So they are very, very important. They are in fact controlling the very development of the body of a eukaryote. So it is so important that even the DNA sequence which encodes for homeodomains has been named called homeobox. These are the sources that I have used and I quote them all here to prepare this lecture. And with this joke, I thank you all for listening. Until we meet for the next lecture, bye bye.